Assalamu alaikum everyone. So today's topic is calcium homeostasis and onwards. So what is calcium homeostasis? Now we should know that plasma calcium levels, they exist in three forms basically. One is ionized or free form, that is Ca positive 2, which is an ionic form, or that is a free form, and that occurs 45%, and that is actually the active form, right? Some of them, 40% of it is bound to the albumin, which is a protein in the plasma, and 15% of them is bound to anions, right, the negatively charged ions. So this ionized or free calcium, as I already have told you, is an active form. So it's a primary regulator of PTH, that is parathyroid hormone. Right. I already have talked about PTH in detail in the previous topic. So you should remember that changes in the pH, that is acidity or alkalinity, that will alters the pH secretion, PTH secretion. Right. I'll tell you, in a moment. Whereas changes in albumin concentration, that will not affect it. Right. Now should remember that calcium competes with the proton, right, H positive ions, to bind with the albumin. So whenever there would be increased pH, right, that means whenever there is alkaline environment, which means less H positive ions, right? So in an alkaline environment, there will be less H positive ions, so albumin will bind easily to calcium, right? And then it will result in decreased ionized, that is free form of calcium. And when the free form of calcium will be decreased, which is an active form, the patient will suffer from cramps or pains, paresthesias, that is pins and needle sensation, carpopedal spasm, right? So that all thing will result, the decreased calcium is a stimulator for increased PTH secretion, as we all know that. Now whenever the pH is decreased, that is more H positive ions are there. So we already have taught, I already have taught H positive ions will basically compete, right, with calcium to bind with albumin. So H positive ions are more, so they will bind to albumin. Instead, calcium will be present more in the blood, that is increased ionized form of calcium will be present in the blood, and that will lead to decreased parathyroid hormone secretion, right? So this way basically acidity and alkaline environment has effect on PTH secretion. Next one is calcitonin. So calcitonin has got totally opposite, inversely related to PTH hormone, right? You need to you remember this PTH, so the totally opposite function is of calcitonin. So first of all, calcitonin is released from which cells? Parafollicular, or the C cells of the thyroid, right? And then it opposes the action of PTH, right? And it is not important in a normal calcium hemostasis, right? And the function of it is that it decreases bone resorption, whereas you remember that the PTH hormone that increases bone resorption, as I already have told you in the previous lecture. So in it, this calcitonin function is decrease bone resorption, right? And so how it regulates, whenever there is increased serum calcium level in the blood, there would be increased calcitonin secretion, so CC, remember, right? Okay. As you can see in the diagram, there is this peripheral blood and there is calcium present in it. Whenever there is increased serum calcium, so it is sensed by the parafollicular C cells in the thyroid gland, right? And that increased calcium will bind here, on the receptors present on the surface, and then obviously calcitonin would be increased, and what it will do, it will basically lowers the serum calcium levels by inhibiting osteoclastic bone resorption, right? Okay. Next comes the glucagon. Glucagon is basically made by the alpha cells of the pancreas. I already have told you. So, and what is its function? Glucagon's function is opposite to insulin. So, what it does is it promotes glycogenolysis, like it breaks down glycogen stores to get more glucose in the blood. It also forms new glucose via gluconeogenesis. It also forms free fatty acids via lipolysis, like it breaks down complex lipids into free fatty acids. Then also this one is very important, that is ketogenesis, right? The ketone bodies are formed by glucagon, right? And the other one is that it obviously elevates blood sugar levels to maintain homeostasis whenever the blood glucose levels fall too low, that is in the fasting state. So in the fasting state, this hormone is actually being released. So how is it regulated? So glucagon is basically secreted in response to hypoglycemia, that is in the fasting state, and it is inhibited, obviously the opposite, that is hyperglycemia also. Somatostatin is an inhibitor. I already told you it inhibits growth hormone as well as insulin glucagon. Insulin, yes, it inhibits glucagon secretion. Amylin also inhibits it. So you need to remember this. Then I have remembered it like this: the glucagon, it has got inversely related to all these hormones like insulin, amylin, somatostatin, right? And the effect of hyperglycemia. So these some MCQs for FCPS part 1. Glucagon is stimulated by, number 1, exercise. like primarily by exercise. Other things are also would be there in the MCQ, but primarily by exercise. And whenever there is increased chronotropic or inotropic effect of the heart, hypoglycemia will lead to prolonged QT interval, right? And the cortisol is necessary for the action of glucagon. These some of the important points. So the next topic is insulin, which is a very famous hormone. You need to remember it's very high yield topic. So it is synthesized. First of all, there is something which is called as pre-pro-insulin. It is synthesized in the ref endoplasmic reticulum of the pancreatic beta cells, right? And then the cleavage of the pre-signal will result in the formation of pro-insulin. The pre part is cut and pro-insulin is left. It is now stored in the granules, right? Now its cleavage would basically will cause exocytosis of the insulin and C-peptide. Two things will be released as shown in the diagram, insulin and C-peptide, right? Now they both are formed equally. Both exocytosis occurs and both of these are increase in endogenous insulin secretion. For example, when there is type 2 diabetes mellitus, right? And when there are insulin secretagogues are given in the patient with diabetes mellitus, and whenever there is insulinoma, that is tumor in the of the insulin releasing insulin, whereas exogenous insulin will lack C-peptide. This is very important concept to remember. That's why in a patient with type 2 diabetes mellitus or type 1, we actually also do this lab, that is C-peptide levels in the blood. So insulin is synthesized in the pancreas, okay, and cleared by both liver and the kidneys. Okay, next is that what are basically its function. These are very important. It will bind with insulin receptors. Which type of receptors are those? Those are tyrosine kinase activity, right? The receptors with tyrosine kinase activity, which is very important. And now it will induce glucose uptake via carrier mediated transport into the insulin dependent tissues and gene transcription. These were some MCQs, but the important anabolic effects of insulin are, so it is basically an anabolic hormone which is making of the things like insulin is an anabolic hormone, and effects include number 1, increased glucose transport in the skeletal muscles and adipose tissues, right? Obviously we need that more glucose in the blood to be consumed by the cells, so it's taken up by the adipose tissues and skeletal muscles. Increased glucose transport. Also more and more glucose, the leftover would be converted into glycogen to be stored, and then of course that would be triglyceride synthesis would be increased within the kidneys. The sodium retention would be increased, right? Anabolic hormone is means that protein synthesis would be increased within the muscles, right? Increased cellular uptake of potassium and amino acids. This is a very important thing, the increased uptake of potassium by the cells. And glucagon release would be decreased. Obviously it's the opposite hormone of it. And lipolysis would also be decreased because we're making lipids now, so its breakdown is decreased. We are making lipids, like that is triglyceride synthesis. So unlike glucose, insulin does not cross placenta. Remember, in mothers with diabetes, excessive glucose can cross placenta and increase fetal insulin, right? In diabetes only. Okay. Then comes that there are some of the transporters of glucose transporters. Some of them are dependent upon insulin and some are not dependent, right? So the ones which are dependent is very famous GLUT4 receptors transporters. These are very important, and they are present in adipose tissues in our striated muscles. Exercise can also increase this expression of this GLUT4, right? Now the ones which are insulin independent transporters, like we don't need insulin for the transport of glucose, because it's emergency situation, like the RBCs, like the brain, the cornea, the placenta. These are very important, and they have GLUT1 transporters on it. These are also independent, and all of these are independent except 4 is only the dependent upon insulin, right? Okay, so the ones which are independent, number 1 is GLUT1, right? In these GLUT2 are basically 2 means bi. So by me we have beta islet cells of the pancreas, liver, kidney, GI tract, right? Then the GLUT3 includes brain and placenta. In the GLUT5, remember, they are for 5 fructose, and they are in spermatocytes, GID tract, right? And then there are SGLT1 and SGLT2 inhibitor, that is sodium glucose cotransporters. They are present in the kidneys and small intestine, we know that. Then brain prefers glucose, but that can also use ketone bodies during starvation. And also these are some high yield points. RBCs utilize only glucose. As why? Because they lack mitochondria for aerobic metabolism, so they utilize only glucose, right? So this was basically the mnemonic for insulin independent glucose transport. These all things break lips, right? These all I already have told you. So the next thing is the regulation of glucose, or insulin, right? So glucose is a major regulator of insulin release, one thing. Now yeah, and increased insulin response with oral versus IV glucose is due to incretins. For example, they are glucagon like peptide 1, right, or GIP, which are basically released after meals and increases the beta cell sensitivity to glucose, right? And when they are released, their release is decreased by alpha 2 receptors and increased by beta 2 stimulation, right? So 2 regulates insulin, right? These two, the alpha 2, beta 2, right? So that is basically regulation. Now this is basically very important. You can actually see in the picture what is shown, that how it is basically there is insulin dependent glucose uptake, and then there is insulin secretion by the beta cells in the blood. So look here, insulin dependent glucose uptake. First of all, this glucose is entering into the cell via GLUT4 receptors. We already know that, right? This is beta cell of the pancreas, right? Now what it will do is, number one, this GLUT4 vesicles are containing this GLUT4, and the insulin is binding to the receptor, which is tyrosine phosphorylation. I already have told you this thing firstly, that the insulin first of all comes and binds with the insulin receptor, which is the tyrosine kinase which has got this activity. It will induce glucose uptake by the GLUT4 receptors, which are carrier mediated, into the insulin dependent tissue, right? And that will basically cause gene transcription, cellular growth, DNA synthesis, and also the anabolic hormone being so glycogen, lipid, and protein synthesis, right? Then the next step is basically what is the insulin secretion, how it occurs by the pancreatic beta cells. So first of all, glucose is a major regulator. We know glucose will enter into the beta cells, right, via the GLUT2 receptors, right? Now glycolysis will occur, glucose metabolism, increase ATP would result. Increase ATP, what this ATP will does is that it will close those potassium channels, which are also the target of sulfonylureas. You need to remember this. These potassium channels these are ATP sensitive, so they will close. Whenever they are closed, there will be depolarization of this membrane, beta cell membrane, and the voltage gated calcium channels will then open up, right? So these calcium channels will called calcium influx, right? Remember this one, this voltage gated calcium channel will get open, and that will increase intracellular calcium, and that will basically is responsible for exocytosis of the insulin granules. Look, this insulin is out and into the blood vessel, right? So this was the mechanism of how insulin is secreted by the pancreatic beta cells. Now at the top I have written some important points, like for FCPS part 1, immediate function of glucose, if comes, then it is glucose entry into the cells, or pH increase and potassium entry into the cell. Then the delayed actions are 2. Intermediate is protein synthesis, and the delayed effect of this is basically lipogenesis by messenger RNA, right? So now adrenal steroids and congenital adrenal hyperplasia. This is also a very important topic, and so I'll start from here. Cholesterol. This cholesterol in our body is converted into pregnenolone with the help of an enzyme which is cholesterol desmolase, right, which has got positive effect by ACTH. So this cholesterol desmolase is a rate limiting enzyme, and this step is also rate limiting. You need to remember this. Cholesterol is now being converted into pregnenolone, number one thing, and then the other series will start to make all the 3 things in the adrenal cortex, the 3 hormones from zona glomerulosa, mineralocorticoids, fasciculated glucocorticoids, and reticularis androgens, right? So this is how we'll actually start. So this pregnenolone now, right, these are basically the hormones which are enzymes, and these are the hormones. So pregnenolone is converted into now progesterone, right? Now there is this one series. First I'll complete this. Progesterone will then convert it into 11-deoxycorticosterone with the help of enzyme 21 hydroxylase, right? Now this 11-deoxycorticosterone is converted into corticosterone with the help of 11-beta hydroxylation. Now corticosterone is finally converted into aldosterone. Remember this. starting like on, right, with the help of aldosterone synthase enzyme, which has got positive effect by angiotensin 2. And this aldosterone is finally responsible for causing sodium retention and potassium secretion. We know that by zona glomerulosa cells it is secreted, and this is also called as mineralocorticoid, right? The next one is that pregnenolone was also being converted into 17 hydroxy pregnenolone with the help of 17 alpha hydroxylase, and this one is when then converted into the same progesterone derivative, and then into the 11 deoxycortisol, and then into cortisol, right? We know that, which is a glucocorticoid, and then into cortisone, right? So this thing, and then again this 17 hydroxypregnenolone is also converted into DHEA, which is dehydroepiandrosterone, which is converted into androstenedione, then into testosterone, right, in the males. And these are the androgens. Testosterone is androgen, and the estradiol is androgen, right? So then what happens is this androstenedione with the help of aromatase enzyme is converted into estrone, and also this testosterone is converted into estradiol, right? And this testosterone in the presence of 5-alpha reductase is being converted into dihydrotestosterone, right? And then finally we have this estrogen and DHT, we know, in the peripheral tissue, and these are the female hormones, and these were the male hormones. These are the androgens, right? So why this was important to remember, because we have got this table to memorize, right? It's not that difficult, but many people find it difficult. So let's get started and make it really easy for you. So first of all, 3 basic hormones are there, right? 17 alpha hydroxylase. First of all, let's talk about 17 alpha hydroxylase. So इस table में what is there, 3 basic hormone deficiencies are very important. Sorry, enzyme deficiencies. Number 1 is 17 alpha hydroxylase. When this enzyme is deficient, we will have mineralocorticoid only in excess. Other cell lines will be decreased. I mean other hormone lines, that is cortisol and sex hormone, they will be decreased. Only one thing is increased, that is this pink one, mineralocorticoids. How? We'll see in the table. This is 17. Wait. This is 17 alpha hydroxylase, right? When it will get deficient or decreased, then what will happen? There's absence of this enzyme. So all the other lines, that is, it is arrowing above ahead. This line making cortisol, and the other one sex hormones. These all the 2 lines will be decreased, while absence of this will lead to increase in pregnenolone. The precursor would be increased, and this cell line will be increased, that is making aldosterone. So its main function is sodium retention, potassium secretion. So due to this sodium retention, BP would be very markedly high, potassium will be decreased, other cell lines will be decreased, as I already have told you. Labs may androstenedione would be decreased. This is that the precursor of sex hormones. So yeah, in XY males, atypical genitalia and undescended testes would be there, while in females, lack of secondary sexual characteristics or development. Right. The next is enzyme is 21 hydroxylase. Right. Now we will see. First of all, remember, in 21 hydroxylase, all the cell lines would be decreased except for the sex hormone one. Right, that would be the only one which is increased. How? 21 hydroxylase. Let's chase it. This one is 21 hydroxylase enzyme. So when this is absent, so this line, this line would be decreased. Also, the other line would be decreased. Only this is leading to a horizontal pathway, like this progesterone will be increased, and that will lead to increase in this thing, and then androstenedione, and then testosterone and estrogen. The sex hormones would be increased only in this, right? So yeah, sex hormones will be decreased, all other would be less. The other one is labs. Labs may there will be increased renin activity and increased 17 hydroxyprogesterone levels. Presentation will be the most common presentation in infancy is the salt wasting, right? Because aldosterone levels are decreased. Or in childhood there will be precocious puberty, and in females there will be virilization. So the next hormone, that is 11 beta hydroxylase. In this also only the sex hormones will be increased, all other will be decreased. How? Look at this. This 11 beta hydroxylation. This 11 beta hydroxylase would be inhibited. So that would lead to only the formation of this androgens, like they would be increased in number, while all the other lines would be decreased, right? Just like this, right? So it presents in infancy with severe hypertension, right? And then there would be childhood, there would be precocious puberty, and you need to remember one thing. that all these congenital adrenal enzyme deficiencies are autosomal recessive. One thing, and are characterized by skin hyperpigmentation due to increased MSH production, that is melanocyte stimulating hormone, which is also produced when there is more ACTH, and bilateral adrenal gland enlargement would be there due to this more ACTH stimulation, right? Remember that on the top of this, I told you that this whole thing is stimulated by ACTH, right? So MSH also. And if the deficient enzyme starts with 1, right, it starts with 1, and that causes hypertension. Like this is 1 starting from 1, this is starting from 1, they both will cause hypertension. While if it ends with 1, then it causes virilization in females. Like this one, 21 hydroxylase deficiency will cause virilization in females. Okay, just a little mnemonic for to remember this. Okay, so this was all about some of the hormones and the details in endocrinology physiology module. So next we will discuss about the cortisol levels in the next video. These cortisol and much more, right? So stay tuned, subscribe to my channel, and also share with your friends. Thank you so much. Allah Hafiz.